Well, my computer says it's 1:01, so I apologize for starting late. But hi, everyone. My name's Cassie Sofing, and I'm with the Globe Mosquito Habitat Mapper app and the Globe Mission Mosquito campaign. Today, I'm really excited about our topic. I can't wait for you to hear our speaker. And without further introductions on my part, because Dr. Lowe is going to jump in on this one.
Oh, great. Oh, thank you so much, Cassie. It is great to be here. And it is even greater to introduce someone that was once a colleague many years ago, but we have since become colleagues and very close friends. And with those words, I'd like to introduce Anita Schiller. Anita Schiller is a industry leader in mosquito control. She has been devoted the past, I don't what, couple decades. She still looks very young, but she's very accomplished. She has done a lot of work with mosquito control in her home in Texas. She has run insectaries. She has been working with different kinds of species, thinking about biological agents in mosquito control. And she's just an all-around superior scientist. And we are just so lucky that she has time today to talk to us about mysterious mosquitoes. So with that, Anita, I just want to give you my heartfelt thanks for you to take time out of your busy day to meet with us today.
Thank you. Thank you so much, Rusty. Wonderful introduction. I'm so honored to speak to you guys today, and I'm absolutely honored to be your friend and colleague. So I'd like to go ahead and get started. Let's see. So today's program is titled Mysterious Mosquitoes. And the purpose of the program is to give you additional information as citizen scientists as far as using the mosquito habitat mapper protocol goes, as far as mosquito biology, larval identification features, some virus interactions, what actually happens inside a mosquito when a virus is taken up, and just all around background information. My name, as Dr. Lowe said, is Anita Schiller. And to give a little bit more background, is that I've spent the last 25 years as a full-time naturalist and then a research scientist that produced and utilizes biological control agents against medically significant mosquitoes in my home turf, Harris County, Texas. The biocontrol work and the biocontrol agents that we produced were used in Harris County public spaces, parks, and preserves where conventional mosquito control just wasn't feasible. That work is actually published in peer-reviewed journals, so you can look it up if you're interested. And as always, I continue to serve as subject matter expert on biocontrol for mosquitoes and mosquito mass rearing methods. So, as I mentioned, this program is in support of Mosquito Habitat Mapper, specifically the GLOBE Mission Mosquito protocol, where citizen scientists provide velocity, volume, value, variety, and veracity of the data to better understand mosquito population range and distribution across urban environments and land cover types. The modelers then use the ground-based mosquito data that you provide to complement and validate the environmental data from satellites and create and improve predictions of disease outbreaks. So your contributions and observations will actually help create new maps that have a finer spatial resolution that would be possible or that is possible using satellites alone. So here is what the Mosquito Habitat Mapper protocol or Globe Observer protocol looks like on your cell phone after you download the app. So there are 4 different protocols, and we're specifically talking about the second one there, Mosquito Habitat Mapper. In my opinion, that's the best one. It's the most detailed, the most detailed, most science heavy protocol. But even though it takes a little bit to learn about the things, so the learning curve is a little steeper than the rest of the protocols, it is well worth it for everyday citizen scientists to learn something new, also for students and teachers to use in their classrooms, and for students to get extra credit in their biology lessons or biology classes. All right, so why are we talking about mosquitoes at all? Why is NASA talking about mosquitoes besides studying them or tracking land cover types from space? Is that overall, globally, mosquitoes do present a huge burden in public health and are considered the world's deadliest animals. So that goes beyond or in front of deaths occurring from homicides, but also from other animals. And biologists actually estimate that mosquitoes have killed about half of all humans that have ever been born. And while we tend to think of the topic as a singular organism, the word mosquito as a singular organism, next slide. Mosquitoes actually comprise, or the mosquito world is actually comprised currently of 3,578, give or take, species that have been described to science right now. Of those roughly 3,600, only 243 are potential vectors of pathogens. And the science currently knows of about 130 arthropod-borne viruses that can be transmitted by arthropods or insects and ticks. Of those 243 potential vector species, only 88 are competent vectors that we know of, that scientists know of, that are competent and suitable and able to transmit pathogens that can cause disease in vertebrate hosts. So to sum it up, of the 3,600 mosquito species described, less than 10% are medically significant. And that means more than 90% of mosquitoes are totally harmless or they're simply nuisance biters. Okay, so this slide right here, I apologize, it is outdated. It is from 2018. However, it illustrates the burden that is caused from arthropod-borne disease, or specifically mosquito-borne disease, in the United States from 2018. And the reason it's still 2018 is that was the last time when the Centers for Disease Control published data in that form where all the arthropod-borne diseases were listed together. Now they have a new system and you have to parcel out all the data yourself and then create your own charts. I feel like for the purpose of this program right here, to illustrate the importance that mosquitoes play in our everyday lives, this slide is still shows the impact of mosquitoes. So you'll see on this slide right here that the middle bar, the West Nile virus, is by far the greatest burden of public health concern for us, and then followed by malaria. Although malaria, for the most point, except for a couple of cases last year, is travel-acquired malaria disease that has been brought back by travelers, and then those travelers became sick once they returned to the United States, went to the doctors and were diagnosed with malaria. But the West Nile virus cases have actually been acquired within the United States. And those are primarily Culex quinquefasciatus and Culex pipiens cost. And who and what that is, I'll tell you here in just a little bit. But besides being able to transmit pathogens that can cause disease, we've recently found out that it's actually possible for mosquitoes to also transfer microplastics into our bloodstreams. Isn't that just a cat's meow? So we found that there's over 12 different microplastics that made it into the gut region of 4th instar or juvenile mosquito larvae, and they actually the larvae transported those microplastics into the pupa stage, and then from pupa into the adult, and then passed them on into the egg stage. So the overall accumulation of those microplastics in the mosquito body and the juvenile body itself will delay the emergence of the adult. So that sounds like a great thing. However, once the adults do emerge, they have been observed to be more aggressive in their blood feeding activity. And so now we know, as I mentioned, that not only can we get sick from mosquitoes, arthropod-borne disease, but also we can get microplastics injected. Before we discuss how to curb mosquitoes overall and how to manage them and how to observe them, I wanted to be sure that everybody knows what mosquitoes actually look like. And I wanted to start with the adult animal. So here we have a little quiz. I know you can't shout it out. I won't be able to hear you, but here's a quiz of different adult insects. And if you think you know the answer, go ahead and say it out loud or keep it to yourself. Either way, just click through that slide, please, Cassie. All right. So, well, I'll go ahead and give you the answer. So the top left is midge fly. The top right is a crane fly. The bottom left is also a midge fly, a different midge fly. We lovingly call them bloodworms when they're juveniles, and we feed them to our aquarium fish. And then leaves the bottom right as the mosquito. And if you click one more time, the proboscis is the action item that will tell us what a mosquito looks like as an adult versus the other flies that are in the same orders of flies that look similar, that are about the same size, that also buzz around our heads or enter our homes, but they all lack that proboscis. Only the mosquito has that straightforward pointing proboscis. So as I mentioned, mosquitoes have a proboscis, and they, taking it back a couple of notches to the beginning, they are an insect. And all insects pretty much share these physical characteristics. And I say all with in quotation marks, all adult insects do. They all have a head region, a thorax region, and an abdominal region. That's the green section, the yellow section, the blue section on the graph on the left. What sets the mosquitoes apart from other insects, say from a grasshopper or from a butterfly, is that mosquitoes belong to the order of true flies. And true flies only have one pair of wings, of flight wings. And then they have a little structure underneath the flight wing that is a residual wing that has become stunted and shortened. And it's called a haltere, which is a pretty jargony word, but it's a vestigial wing. And that helps kind of like a gyroscope, to keep the insect flying forward and up and upright without turning circles. So all true flies only have two wings. Then the mosquito itself has scales all across the body, and that includes the wings. And the wing venation and the scales on the wings can actually be identifying features. And then, as I mentioned, mosquitoes have a proboscis. It's usually forward pointing, and most of them are straight, but there are a couple of exceptions, and I'll get to those in a minute. So how do you tell a male from a female mosquitoes, and do you even need to bother telling them apart? So here's an image of two very large mosquitoes, that's male and a female, of Toxorynchides, or adult mosquito assassins. That is the sexy mosquito that I spent most of my adult entomological career focusing on. And on the left, you see the male, on the right is the female. And it's pretty obvious right away when you look at the head region what the differences are. And you can see the male on the left has very bushy antennae. And in a mosquito and most other insects, adult insects, the antennae is where the neuroreceptors are. So it's kind of like a nose for them. And because the male's sole interest is finding a female's, and the female gives up pheromones and cheromones, the male's job is to smell her out. So he needs to have a big nose or big bushy antennae. The female on the right, if you compare her antennae against his, they're relatively small. You can hardly see them at all, but she has large maxillary palps that follow the proboscis, and her proboscis is downward curved because she never takes a blood meal and she only feeds on flower nectar. A medically significant mosquito here still shows the differences of male and female. This time the female is on the left and the male is on the right, but you see on the male having those bushy antennae, whereas the female does not. And this is Culex coronator, a local species, a floodwater species. As far as the mosquito life cycle goes, because we learn to identify adult mosquitoes, we also need to learn the rest of the life cycle, because in Mosquito Habitat Mapper, we're not actually interested in observing or reporting adult mosquitoes. We actually need to learn what the life cycle is and follow that life cycle to get to the juvenile stage. And here you see an example of a mosquito life cycle that uses Toxorhynchides, again, the mosquito assassin. And it shows us that the Toxorhynchides, as all mosquitoes, follows a holometabolous life stage or life cycle, meaning it's a complete mosquito cycle, a complete life cycle. That means that there are 4 distinct and separately looking stages of the same insect. So here you see on the top right a yellow flower with the Toxorhynchides feeding on the flower. Below it, we have eggs that are laid. Then those eggs, the eggs are always laid onto water or near water. When the eggs hatch, we have a larva, and all mosquitoes go through 4 separate instars. And an instar means it's the stage between the molts. Being that they're invertebrates and insects, they don't have bones, they have an exoskeleton. And in order for them to grow and change in size, they need to shed that exoskeleton. So the jargony word is an instar. It just refers to the molt stages of the juvenile. Then the last juvenile phase is a pupa stage, and that is on the bottom 7 o'clock, that round organism there, that's the pupa stage. And if you're not familiar with the pupa stage, the best way to explain it or to envision it is if you're familiar with what a caterpillar life cycle goes through, or butterfly or moth, is that they feed us caterpillars and then they'll build a cocoon or chrysalis. And inside that cocoon and chrysalis, they metamorphose into the adult. It's where the adult body is formed. And that same process really occurs in the mosquitoes as well in the pupa stage. So as a larva, the larval stage feeds and is actually a ferocious, full of appetite larva. But in the pupa stage, they can no longer feed, and that's where the adult body is formed. If you look up close, you can see the trumpets, the 2 little horns that stick up out of the water. After the pupa stage is completed, and it's the duration of the pupa and the larval stages and even the egg stage really depend on the species. They're all different. In Toxorhynchides, it's rather lengthy process. It can take weeks, if not months. And in other species, it only takes a few days. So in the Toxorhynchides phase here, the pupa stage is 4 to 6 days, and then the adult emerges. Once the adult has emerged, again, the male looks to mate and the female looks to get her energy drink. In Toxorhynchides, that's a flower meal or nectar meal. And then the mating takes place, the eggs develop, eggs are laid, and it all repeats. Next. Again, the Mosquito Habitat Mapper wants to or is asking for data as to where the mosquito breeding occurs. So this is what the screen on your phone will actually look like when you go through the protocol. And the first thing that we need to report is what the habitat looks like. So it's asking whether or not it's a still water system, whether or not it's a flowing water system, artificial container, a natural container, whether or not it's a trap that is set for the mosquitoes themselves, or whether it's an OV trap. And you might say, wait, flowing water, mosquitoes don't breed in rivers and flowing water. That's not even possible, right? And if you ask that question, then I have that answer in just a little bit. So still water for the Mosquito Habitat Mapper would look like this, and these are examples. So still water refers to whether or not it's a lake or a pond, and you're not looking for the mosquitoes out in the middle of a giant lake like Lake Michigan or something. You're looking for mosquitoes that are actually attached to the water plants that you see in the top left here, or on the margins along the lake and the pond. Still water can also be stagnant and non-flowing ditches. And if you ever spend any time in Houston, you know Houston has a lot of ditches. So they're actually a big problem in my area. Swamps and wetlands, most people consider them to be mosquito breeding habitats, and they're not wrong. But very often, swamps and wetlands are high in biodiversity with a high load of predators that keep the medically significant mosquitoes down low. Here in the United States, but also globally, you might find mosquitoes in a still water habitat that's inside a vehicle track, like from construction vehicles that are heavy, that sink down in mud. They might leave divots behind where rainwater can collect. And in Africa, even animal tracks, like elephant tracks, can hold enough water in them for a very problematic mosquito to breed in. Salt marsh and bays breeds the Aedes sollicitans or salt marsh mosquitoes. So those tend to not flow, even though the tides will bring water back and forth, but generally the water sits. And then the biggest one are and I say the biggest one, at least for Harris County, for Texas area, are the underground storm drains. Those often get clogged by debris, by trash that gets blown into storm drains. And when they're underground, those clogged storm drains are nicely insulated and create wonderful, wonderful, sarcastically speaking, mosquito breeding habitat. It is such a problem that our local mosquito control district spent several years worth of trying to fight the underground mosquito breeding population with adulticides, with sprays, with fogs, and never could get rid of them all. In the app, the next category in mosquito breeding habitat is flowing water, and that was the one I said, see, the mosquitoes don't breed in flowing water. And if you thought that same, you are correct. But what often happens with flowing water is that it's a natural flowing water, like a river, and rivers tend to move back and forth with the water level. And here you see a perfect example where the river is behind in the background, behind that person, and in the foreground you see that heart-shaped green puddle. So that puddle is left behind after the water recedes. So it's still part of the river system, but at this point it's still water. Artificial containers is not hard to explain. It is something man-made that holds water. And here you see a number of examples, but what they all have in common is that they hold water for more than 5-6 days, that they're all man-made, and that they lack predators. Some of the worst offenders for container breeding habitat are the tires that you see there in the middle. Tires are perfect for mosquitoes because the medium that they're constructed out of is very insulating. The shape allows for the water that condenses to just trickle right back down into the tire well, and the water can stay inside a tire well even during times of drought. Neglected bird baths, folks that like to feed birds and give them places to splash in sometimes forget to clean out their bird baths. And I've seen many of those examples in real life where I had to educate the folks that they needed to really turn over their bird baths and give them good scrubs every week. And what folks tend to forget about is that mosquitoes can actually enter our homes. And if you remember that chart earlier where I showed the mosquito-borne disease burden for the United States, with the highest one being West Nile virus. Well, West Nile virus, the biggest vector for West Nile virus in the United States is the southern house mosquito and the northern house mosquitoes. So they will readily enter your home and look for places to breed inside. The northern and southern house mosquitoes they will breed in any water that they can find. They're not picky, and they will breed inside a toilet water tank that hasn't been flushed in a while, even a P-trap, inside dishwashers that haven't been used and haven't drained. They will breed anywhere inside your home where they can find water. So don't forget to look there as well. Natural containers, as the name implies, are not man-made. They're naturally occurring places, and usually we think of them as tree holes or natural containers, and those are easily observed in the wild, but sometimes folks also create their own mosquito breeding habitat or their own mosquito problem in their own backyard by planting outdoors bromeliads. And here you see on the left in the red, with the red images, those are tillandsias or bromeliads. They're tank bromeliads specifically. And you're actually supposed to water them by watering the crown of the plant that then holds the water. Well, that's great for the plant, but it also creates that natural container mosquito breeding habitat. And here are just a few local examples that I like to throw in. This is literally in my neighborhood area where I don't have to look far for mosquito breeding habitat. This is right in the vicinity, and you probably have similar instances as well. With the top left being right outside my fitness gym, great big old container plantings with saucers on the bottom, and the mosquito breeding habitat is within those rocks. So as the plants get watered on top, the water seeps out on the bottom, gets collected inside that saucer, and that is where the mosquitoes breed. The trash can in the bottom is also an interesting one. Folks tend to not think about trash cans, but if the trash cans don't have drainage holes drilled in the bottom and lack a lid, well, they can fill out completely in a good downpour. If you don't toss them out, you have your own mosquito breeding habitat. And then the other one I'd like to mention here is the bottom left. That is the water meter that's inside a very well and very nicely manicured lawn. That lawn gets irrigated, in my opinion, way too often. And what happens is with all that irrigation runs off right into that water meter and keeps that water meter flooded. And even though the homeowners keep that water meter covered, and you see the cupboard is set to the side in that picture, you see the hole that is used to remove the cover with. Well, that hole is big enough for mosquitoes to enter, and they do find those irrigation meters. All right, so now that we know where to look for mosquito habitat, we'd like to know a little bit about what makes the mosquitoes tick and what allows them to find that habitat. So first off is the breeding. So here you have a picture of a very unadulterated picture of two mosquitoes copulating. You have the female is on the left and the male is on the right. And you see he's somersaulted in order to clasp her. The claspers are on the tips of the abdomen, and his claspers actually stick out. I don't know if you can see me. His claspers stick out, grab a hold of her, and pull his genitalia close to hers before the genetic material transfer occurs. You also see that she is full of blood. She's nice and red. She's fully saturated with blood. And I believe, oh yeah, she's also imbibed right there in the skin of a human. So mosquito copulation involves some somersaults. Next slide. After the copulation takes place, either the eggs have already developed or they've simply and getting fertilized, or the eggs will develop. But the female, depending on the different group of mosquitoes or the different species, have different behaviors as to how they lay their eggs. Anopheles is on the left of the slide, you see her resting on the water surface. Anopheles is the problematic mosquito in the African region African and South American region that vectors the malaria protozoan parasite. So she sits on the water, but then she lays single eggs. The Monsonia in the middle is an oddball. She actually dives and dips her abdomen under floating plants. The floating plants are usually Lemma minor, duckweed, and Lemma minor, Lemma major, and little floating plants like that. So Monsonia will just dip under the water and cement the eggs to the underside of the leaf. Culex will sit on the water as well. So she will rest on the water and actually taste the water first. That's the group of mosquitoes I spoke of earlier that where the northern and southern house mosquito belong to. She'll sit on the water, taste the water, make sure it checks all her boxes before she lays a bunch of clustered eggs. Aedes are the ones that are actually responsible for those all of a sudden mosquito clouds after long droughts, where you say, golly, there hasn't been rain in a long time. Where did all these mosquitoes come from so quickly? Aedes will lay her eggs either at the water meniscus or just above it onto substrate, and that is in an area that is likely to flood again. So her eggs will then sit, dry out, and wait for the next rain. And toxobronchitis on the top, I don't know if it's visible on your screen. She is the weirdest of them all. She actually just baseball tosses her eggs right into a container, a natural container usually. She just releases them in mid-flight and tosses them in. And they're pretty good aims. They can actually hit a itty bitty tiny hole inside a bamboo comb in the tropics. As I mentioned, the mosquito behavior is different from group of mosquitoes to different species, and so are the eggs. So the eggs show a great amount of diversity, with the top left being the Culex type mosquitoes, the Callicinae. They all create a raft of vertically aligned ovi or eggs. So the ovi you see here, they're actually stuck together, and all the eggs have a little bit of texture to them, and that texture kind of works like velcro. And so as the eggs are deposited or oviposited from the female, the hairs on the eggs just interlock and allow them to stay together in a raft. The bottom is what the Anopheles eggs look like. Again, they're laid singly, but they have little air floats to them. So both of those Culex and the Anopheles eggs below them, they have to stay on the water in order for them to hatch. In the middle is the toxobronchites. You see how white they are, and they stay white. The top right are the Aedes eggs, and you see them here laid against a piece of wood that's next to the water surface. So they're single eggs, kind of stuck next to each other. They're truly single eggs. Here they're just laid in a row. And then the Monsonia eggs are the clusters that are under the water surface. So the jargony way to explain the eggs is that they're actually made up of follicular epithelial cells, and the proteins that they produce will slowly scleroterize and cross-link together to harden the eggshell. So scleroterization means they darken with time. They start out white and then they turn black or dark brown. And the chorion is what we call the eggshell in the mosquito egg. As I mentioned already, is frequently sculptured or textured. Here you have the process of that Monsonia mosquito as she dips underwater to lay her eggs. It's pretty fascinating. There are a couple other mosquitoes that do similar things, Cucullitidia, for instance, and all these are bird host blood taking mosquitoes. They also dive underwater and then lay their eggs right into vegetation. And then here is a slow motion video of that odd aerial toss behavior by toxobronchites. And if you could hit the space bar for me, Cassie. I hope you all saw that. It's pretty interesting how that works. Now, this is in the laboratory. This is in a cage with a black cup. These toxobronchites were bred and conditioned to lay their eggs inside these cups, but in the wild, they would breed or lay their eggs inside tree holes, clogged gutters, and anywhere else that is rainwater-filled container that lacks predators. Next slide. So now that we have a little bit of information about how the mosquito works as far as mating and egg production goes and where they lay their eggs, let's see if we know what a mosquito larva looks like. We have another quiz, and again, if you'd like to send up hearts, if you think you've found the mosquito larva in this set of images. All right. So the top left is what we lovingly can call a bloodworm. It's a chironomid fly or a midge fly. They actually produce hemoglobin, and that's why they're red. It's an oddball in the insect world. Again, bloodworms is what we buy in for tropical fish food. Below them are moth fly larvae. Some people call them filth flies. They can be found in your P-trap inside the home or in bathrooms sometimes. The itty bitty little flies with highly scaled wings, and they just look like little tiny teddy bears flying around. The bottom right is one that we generally don't like too much because that one is the juvenile form of a biting midge fly, Ceratopagonidae. They'll breed just about anywhere where there's mud and muck. And Florida has a big problem with biting midges. I think they call them no-see-ums and punkies that come out at sunrise and sunset. So that leaves us with the top right as a mosquito larva. And that one, I believe, is Aedes coronator. All right. So now that we saw what a mosquito larva looks like in real life, let's see what makes the body structure of the mosquito a mosquito. You have here a diagram of two different subfamilies of mosquitoes, the Anophelines and the Chelicinae. We can divide the mosquito problem in the world to these two groups and be pretty optimistic that we catch most of the medically significant species. So on the left, we see the Anopheles. Again, those are the ones that can transmit malaria. And Chelicinae is on the right. What is pretty obvious right away is that the chelicines on the right have a siphon, and that is that black thing on the bottom of the abdomen that sticks out. And if you think of it like a snorkel to breathe underwater with, then you have the right idea. The snorkel sticks out of the water. It penetrates the water meniscus, the water surface tension. And so the mosquito actually breathes atmospheric oxygen, so regular air. They do not have gills. Whereas the Anopheles on the left lacks that siphon, no snorkel present. So they have a different adaptation in order to breathe because they also lack gills. So what they have are these tertial plates that help them stay buoyant, and those are those oval dark shapes on the top part, on the dorsal part of the abdomen. And they also have these palmate-shaped hairs that are also on the dorsal and the side of the abdomen. And with a combination of those tertial plates and those palmate hairs, it actually keeps those larvae parallel to the water surface and allows that itty bitty tiny little stopper at the end to penetrate the water surface and for them to breathe. Chelicines lack those palmate hairs, they lack those tertial plates, but they do have that siphon. So here you see the diagram of what they might look like in the water, and it seems pretty obvious and self-explanatory. However, when we're looking inside the water for mosquito breeding habitat, we might look deep into the water with our eyes. We might scan the bottom of the water. We might look into the water column, but we might forget to look on the top of the water. So here you see what the Anopheles mosquito larva might look like on the top of the water. They very often resemble like a little tiny bit of grass that lays on the top of the water. And it's really easy to see whether it's a piece of grass or a mosquito larva, as you just kind of poke at it. And if it moves away and skitters away, it's a mosquito larva. So, and then you see how the culex and the Aedes use that siphon that sticks through the top of the water surface, and the body actually hangs down into the water column, which is how they feed. So that's an important factor. Anopheles feeds on the surface of the water, and culex and Aedes feed inside the water column and on the bottom. Next slide. Here's a slide with courtesy of Cassie of what mosquito larvae actually look like inside the water column and on the water surface. The long skinny things hanging down are the larvae, often called wigglers because they wiggle around in the mosquito column. And then those round things tumbling around are called tumblers, or the pupae of the mosquitoes. So there are multiple age groups right here, but they're all still juveniles. The feeding modes of the larvae are different as well, where most culicines will stay on the water surface in order to feed, but they move their head back and forth. Kind of if you imagine what a hammerhead shark looks like when it's looking for food underwater. So that's what a lot of the culicines will do, moving their head back and forth and grabbing food like diatoms, algae, little bits of debris, and little ciliates and such, seeds and things that are in the water column, and move that with their comb plates or brush combs towards the mouth. Many of the Aedes will actually leave the water surface and dive down to the bottom of the water container or water area, water habitat, and graze on the bottom. So they're more grazers, they're more harvesters. And then, as I mentioned already, Anopheles actually stays on the surface of the water. Since they lack that siphon, they don't like to leave the surface. And what they do is they're very much like ciliates, where they create a torrent with the water by moving their comb brushes, their mouth brushes around, that creates the current that moves the food to them. So they actually don't have to move around a whole lot to catch their food. They create the current that brings the food to them. And here, if you see the arrow, I believe you can see the really odd adaptation of Anopheles mosquito as to how they're able to just feed on the surface of the water. So I don't know if you saw that, but they're able to turn their head 180 degrees. So they can feed on the top of the water and then turn their head around again to look into the bottom of the water, just to make sure that there's not any predators that come up from below. So I always think of that as like the Blair Witch thing. She can turn her head 180 degrees. After the larval stage comes the pupa stage, and that is the same for all of the mosquitoes, Anopheles, Colocines, Aedes, Toxomichides, and all. Again, that's where the metamorphosis takes place. No feeding takes place, and this miraculous thing changes from that long, skinny, wiggling larva into the adult. So the interesting part here is that during the pupa stage, all the energy needs to go towards the metamorphosis. So they don't have an ability to feed, and they want to conserve the energy, so they become buoyant by trapping an air bubble inside their body. The air bubble is in this light structure, that S-shaped structure inside the body next to the legs. You can almost make it out on this picture. And then they breathe still atmospheric oxygen, so regular air, with these two trumpets. So before, the siphon was on the back end at the abdominal end in the larva. So now in the pupa stage, they breathe through an area that are spiracles located near the head. So those we call trumpets. And they get around by just having a jerking muscle contraction that allows them to tumble through the water, and they get around pretty quickly, and they're able to evade predators rather well, actually better as pupae than larvae. There's a couple of predators, biocontrol predators out there in the world that actually know this, and one of them are the water striders, and they will feed on pupae. Well, sometimes they will gang up on the pupae with having multiple water striders, and whirligig beetles too. If you ever look on a pond and you see a bunch of whirligigs and water striders, they'll be in a communal and an aggregation, many of those insects together roaming around on the water surface, and they'll wait for those pupae to sit still for a minute before they'll nab them. And then there's an interesting biocontrol agent that's actually a flatworm that is a specialist on predating on pupae mosquitoes. Unfortunately, this particular program is not on biocontrol agents, so I don't really want to go into the details of that, but I would love to be able to show you all, maybe in another webinar someday, the details of the biocontrol agents. Anyway, moving on, next slide. All right, so now we've gone through the water process, and let's just imagine that the pupae is fully developed, the teneral has come out of the water, and now we need to see whether all of our mosquitoes actually are a problem. If you remember earlier, we said that out of the 36 roughly 100 species, there's only less than 10% of them create a problem. And here is why. So whether or not a mosquito bites can put them into three different categories. The anatogenous ones are the ones that always bite. That means they generally grow up in a water that's nutrient poor. They feed on algae and detritus and things, little bits of leaves and what have you, but that area is not protein rich. the teneral comes off the water, and in order for the follicles that have developed in that female body to turn into eggs, she requires some amino acids. Well, she doesn't have those amino acids stored because she didn't get them in her juvenile nursery water. So she needs to get them, and she gets them via a blood meal. And that blood meal is, she'll extract the proteins and the amino acids, and those will then contribute to building her eggs. There are other species that can come off the water with the first batch of eggs already developed, but then they'll have to take a blood meal afterwards to create more clutches of eggs. And then the autogenous ones, or the real bizarre ones, that's very, very few of them. It's just a handful of species that never take a blood meal. They're very often predatory in their larval stage, meaning they feed on other organisms that are rich in proteins. So they have all those lipids and those proteins already built up in their system, carry it over into the teneral, into the adult, and are able to lay their eggs without requiring a blood meal. And very often they even cannot take a blood meal. Very often they're adapted, their proboscis is shaped in such a way where they can't even pierce skin of any kind in order to take a blood meal. Toxorinchides, that we call the mosquito assassin, is one of those. And I have a few other examples later on. Here's the life cycle again of the mosquito assassin. I mentioned they are predatory in their larval stage. And if you're interested in mosquito assassins, you may have them in your neck of the woods. They occur throughout the entire world, mostly in the tropics and the subtropics. Here in the United States, we have a couple of different species. We've got Toxorinchides rutilus and septentrionalis, and we have Toxorinchides moctezuma in the very, very southern part of the United States. Southern part of Texas and southern part of Florida. And then Toxorinchides rutilus septentrionalis occurs along the central to eastern side of the United States. So to find them, you might look for natural habitat like these tree holes, and for white little balls floating around, skittering around. Again, the larval stage takes 4 stages. But in each stage, they're aggressively predatory, and they'll feed on anything, including their cohorts, including their siblings, anything that triggers feeding response by touching their sensitive setae on the side of their body. So that those little hairs are like trigger hairs, kind of like they're sensitive, like kitten whiskers. So when something moves around and they feel the vibration, they'll turn around and grab whatever that is and feed on it, like you see in that picture of a mosquito assassin feeding on a culex. What's interesting in mosquito assassins as an autogenous species, again, they don't take a blood meal. They cannot take a blood meal, and you can see that on that downward curved proboscis at the end there. But they do require proteins in their adulthood, and they have figured out that extrafloral nectaries, so the nectar, the sugary nectar juices that are produced by plants and tree sap, and floral nectar itself is actually high in proteins. And so that helps them create extra clutches of eggs. They can also overwinter in an insulated area as the 4th instar larva. They can actually starve for several weeks, if not a few months. And in the summertime, if they're in the south and it hasn't rained for a while and it's a drought, they can actually go into quiescence and not come off the water. So they'll just suspend their animation. And then as the temperatures gradually get a little cooler, from the hundreds down into the 80s and 90s, the development will commence and they'll come off the water to fly. Another interesting autogenous one, and I wanted to show this one. You probably won't find it very often in your mosquito habitat mapper protocol searches, because the area where these are occurring is pretty limited to the southern and coastal areas of the United States. These are the purple pitcher plant mosquitoes, or Wyomia smithii, and they're interesting in multiple ways. First off, they're inquilines, and they're specialists to the purple pitcher plant. They don't grow anywhere else, this particular phenotype here. They don't grow anywhere else. So a purple pitcher plant is a carnivorous plant. And by definition, the carnivorous plant actually consumes or attracts, predates, and consumes insect prey that falls into its pitchers. But the Wyamaya smithii has become immune to those enzymes and can actually live inside the waters that are inside the pitcher plant. So inside the pitcher plant, the Wyamaya smithii will feed on the insect carcasses that fall in and get digested by the plant. They'll co-feed on them. They'll feed on another inquilina midgefly that also lives inside there. And what's interesting is that this Wyamaya has actually figured out that they don't have to fly off. They don't have to fly away to get a sugar meal as adults. They can just feed on the sugary substance that the pitcher plant exudes that attracts the other insects that then slip and fall into the pitcher. But the Wyamaya will just sap that right up. What's interesting about this one is that, again, it does not take a blood meal, but only in the population that occurs on the northeast, where the purple pitcher plant occurs. There is another phenotype, and there's actually quite a bit of scientific interest that's going into answering the question as to whether it's a phenotype or a different genotype, a different species altogether. But for now, we're calling it a different phenotype in Wyamaya smithii that's in the south, in the Florida area, that actually grows up in bromeliads that are on trees, and that one will take a blood meal. So it's an interesting adaptation from an academic standpoint. All right, so now we know that some mosquitoes take a blood meal, some don't, and you can often tell whether or not they do by looking at the proboscis. But that's really hard to tell because you have to get really up close to the mosquito. If the mosquito is alive, they tend to fly away, right? In the case of Toxoencetes, if you see them, they might be on a flower and you can get a closer look. The one in the middle there, Malaria, looks like a golf club, and that also is a mosquito that's autogenous. They cannot take a blood meal, and I'll show you some interesting facts about that one later. But the vast majority, so 98% or 99% of all mosquitoes do take a blood meal of some sort, and they all have that forward, straight pointing proboscis. Next slide. So the proboscis, if you think about it, is a pretty dainty adaptation, a dainty thing that the mosquito has. If it was to fly into, say, a window pane, it might break off. So it has a protective layer, much like a sword. The labium, or the main part of proboscis, actually has a sheath around it. And you can see on the picture of the right side, when the mosquito pierces the skin, that sheath gets pushed backward and pushed out of the way. So then the mandibles and the maxillae come into play, and this happens very, very quickly. The mandibles will spread the flesh apart, and the maxillae will create a channel for the labium to push further forward. Again, this happens within a couple of seconds for that flesh to be moved and spread apart. The next step is that saliva is injected into the vessel, and that keeps, of course, the blood from coagulating. So this is a very important factor to remember on how disease transmission actually occurs. And you see on the left diagram here where that happens. So you have the hypertharynx, that's the jargon word for the saliva channel, where the saliva comes out, and the labium in the middle, in the labellum, where the blood gets sucked in. Okay, so what happens to the blood? Remember, the saliva gets pushed out, keeps the blood moving. So then the blood is sucked up. But what happens to that blood? So the interesting part is that the mosquito is able to differentiate between all the sugars and what it takes up, like the floral nectar that it sucks up, and the sugars that are in the blood. And it divides it up. The carbohydrates, the sugary stuff, gets moved into a part of the gut that's separate, and we call that the diverticulum. But then the blood, the business part of this thing, is actually pushed on to the midgut. And that's where the magic happens. And I say magic because literally, I feel like the universe just aligns itself for a virus to do what the virus needs to do. So if you imagine a female mosquito having taken a blood meal from a vertebrate that has an arbovirus in its blood, and we call that a viremic host. Let's say a person has dengue, is sick with dengue, and an Aedes aegypti comes along and takes a blood meal from the person with dengue. So the person with the dengue is the viremic host. The mosquito takes the blood meal, and the blood travels into that midgut, as I discussed in the previous slide. But there, a couple of things happen. If it's a mosquito that's not Aedes aegypti and a different, say a Culex of some sort that's not compatible with the virus, then the virus gets digested. It gets inactivated. But if it's a mosquito that has co-evolved alongside this virus and vice versa, then the magic happens that the virus can replicate inside the midgut. And once it replicates enough, it can leave the midgut and kind of break the jail cell, if you will, of the stomach and start to populate the rest of the body and just keeps building more and more copies of itself. It replicates and replicates, fills the entire body of the mosquito until it ends up in the salivary glands. And remember how I said earlier that was important, that first thing during a blood meal, the mosquito spits up a little bit into the vessel to keep the blood moving? Well, if the virus is in the salivary gland, then the mosquito can pass on that virus. But it has to be the right species of mosquitoes meeting the right virus and vice versa. Then there's also a question of time. The replication of the virus just takes time. And that time can actually be measured. And when we measure that, we call that period of time the extrinsic incubation period. So before that virus gets to the salivary gland, if the mosquito takes another blood meal, the virus hasn't made it there, it cannot pass on that blood meal. Okay? But once that time period has passed, the virus can be passed on. That extrinsic incubation period, or EIP, is kind of dependent on the virus itself. And so we can study that from an epidemiological standpoint and a mosquito control standpoint as well. From the time the first virus was detected in the mosquito population as to when the mosquito control has to happen before people could potentially get sick. But in the epidemiological world, that, like I said, that is really like the universe has to align, but we can again map that out. And so what happens is that whether or not a person or a host, an animal, becomes infected with that virus really depends on whether they're in the right environment, whether or not it's the right pathogen that's suitable to infect the special vector species, and whether the vector species is present, and whether or not they're all competent for each other. So, for example, using a Cholecida melanura, which can vector Eastern equine encephalitis in North America, they usually take the blood of small birds. And the birds are the main reservoir of the Eastern equine encephalitis. And that things are fine as long as they stay in the birds. So early in the season, while the mosquito numbers are relatively low, the birds are not really bothered by a few mosquitoes here and there. But as the mosquito numbers, or the Cholecida melanura numbers, rise, the birds are then forced to actually defend themselves, and they defend themselves to such a point where the mosquitoes becomes less selective of the host. And then soon they'll move away from the birds and start attacking mammals as well. And that becomes then the Cholecida melanura then becomes the major vector of the Eastern equine encephalitis virus in the United States. So you might say, well, man, it hasn't rained for a long time. We are in the clear, right? We don't have to worry about putting on insect repellent because it hasn't rained. There's no mosquitoes, right? Well, you're actually, if you think of it that way, you're actually in the wrong. When it hasn't rained in a long time, you need to be extra, triple vigilant, because drought in truth does exacerbate mosquito-borne virus transmission. And how in the world does that work? It sounds like a paradox, right? So in the water, the young mosquitoes, they're really vulnerable to being predated upon by dragonfly larvae and fish and other predators. And when adult females mosquitoes choose where to lay their eggs, they use their sense of smell in their tarsi, in the ends of their feet, to detect the predators, and they often attempt to avoid them. So, along with eating the mosquitoes, the predators will also then reduce the mosquito population in other ways. So, predators can disrupt the feeding among young mosquitoes, and that may affect the time that it takes for them to grow into adults or the size of their bodies once they reach the adult stage. And although the impacts of those different predators have been tested separately in multiple settings, the overall effects of predators on the ability of the mosquitoes to spread disease to humans really remains unclear because it's really difficult to track. So to address that question, Russell Herzog et al. Used an approach that they call a meta-analysis on the data from previous studies. And they found that along with increasing death rates of mosquitoes, the presence of predators also leads to a reduction in the body size of the mosquito that survive. And that causes them to have a shorter lifespan. And for that reason, they then produce fewer offspring. And then they also found that one type of mosquito, like the Culex pipiens and Culex quinquefasciatus that carry West Nile virus, actually avoids laying its eggs near the predators. So during the droughts, the increased predation in the streams, ponds, and other water aquatic environments will actually lead the female Culex mosquito to travel away from the few bits of water that they came out of before as youngs, and choose to lay their eggs in areas that are closer to human residential areas that have fewer predators because they're usually more developed and less biodiverse. So that is really an interesting concept as to why an outbreak of West Nile virus in humans is more likely to occur during a drought. Malaria actually made a comeback to the United States last year as a locally transmitted pathogen. It was eradicated from the United States after the 1960s or 70s, considered gone from the United States, but as a symptom from a COVID response and redirecting public health mosquito control measures towards COVID response, less mosquito control took place, and as a result, we saw malaria pop back up, luckily in only a few little places, although one of them was also in Texas. But what was interesting on malaria, the malaria parasite, is that it really goes back to dinosaur times. And just a couple 2 years ago, we actually got proof of that this primitive mosquito here that you see on the slide is an Anopheles mosquito that goes back to about 100 million years ago. You can tell it's an Anopheles and not a Culisine mosquito because of the maxillary palps. So if you look at the proboscis or the head of this mosquito, you see there are multiple lines next to the proboscis. So you might think that's a male, but the scientists really know that that's an Anopheline by those lines. Those maxillary palps, they lack a few indicators for it being male. So they're Anopheles. So this was found in amber. And by looking at the Earth timeline, we can tell that 100 million years ago was just dab smack in the middle of the Cretaceous area. So if you are old enough to have seen Jurassic Park, the original Jurassic Park, you might remember that the grandfather and inventor of Jurassic Park in the movie carried around a staff that had a big old chunk of amber on the staff with supposed mosquito inside it. And his premise for Jurassic Park was that he extracted DNA from mosquitoes found in amber and was able to splice the DNA into living frogs to create us dinosaurs. And for 30 years, scientists said, oh, that is so much baloney, that it's all movie magic, because we know that mosquitoes were not around when dinosaurs were. They only started to come around until about 45 million years ago or so, is what we thought. And then along comes the scientist a couple of years ago with his new find of amber. And of course, that mosquito put it at 100 million years ago and proved a scientist wrong once again. So Michael Crichton was right insofar as saying that he could have possibly extracted DNA from the amber mosquito and built his dinosaurs. And I'll leave it as that. So that's a really long time for viruses and for organisms to co-evolve. And the mosquitoes themselves have co-evolved as parasites, so to speak. And you can see that on this phylogenetic relationship graph here on the left, where you can see different types of mosquitoes and how they co-evolved alongside their host preference. So some of the mosquitoes are really specific as to where they will take their blood meal. And you can see the midge, the chabarus, is an outgroup that's on the very, very top. It's an outlying but distant related tiny fly. And so the taxa here color coded by their dominant host. And you can see on the very bottom is that Wyamaya smithii, and it is listed as an obligate non-blood feeder in the northern part of its range. And then because there's multiple color bracts on that chart on her line, you remember that part that in the southern part of the United States, Wyamaya will also take a blood meal. And then there are other examples here as well. There's some mosquitoes that will take a blood meal only from amphibians and only from reptiles. There are Sapphirinas that will take blood meals only from leeches and earthworms. Then there are a few that only take blood meal from small mammals. Those will then transmit St. Louis encephalitis and La Crosse virus. And then we have generalists that will take a blood meal from anything and anybody. But since we're not anybody and anything, we're humans, we're often, well, geez, what makes us so attractive to mosquitoes? What makes me attractive and my neighbor not at all? I know of people that say they've never gotten bitten by mosquitoes, and other people say they're really mosquito magnets. And in short, is that we don't really have a definitive answer for that leads us to one single thing that we could disrupt. It is a combination of attributes that makes us attractive. But the biggest one by far is the amount of carbon dioxide that we emit when we breathe. So the harder we breathe, like through exercise, the more attractive we become to certain mosquitoes. Scientists have also figured out that O blood type is highly attractive to mosquitoes. So if you have that, you better be sure and use extra insect repellent. And then a decreased skin microbiome, so very clean skin that's that lacks a diverse microbiome, is also to blame for mosquito attractiveness. Meaning, there's some microbes on our skin that feed on microbes that produce volatile compounds. So volatile. How do you say that? Volatile compounds that come off the bacteria as a byproduct. So the cleaner we are, the fewer biomes we have on our skin. And then when we sweat, we also produce lactic acid. And pregnancy is also to blame. And there's one more thing that's really interesting. If you drink a lot of beer, the byproduct from your sweat also makes you more attractive to mosquito. You might say, geez, all this disease transmission, what good are mosquitoes after all? Is there any redeeming attribute that a mosquito really has? Well, there are a few scientists that have looked into that. And I'm happy to report that there are a couple of instances where mosquitoes are actually the pollinators of plants. And to prove whether or not that's true, here we have some images from a study that show that this one is Culex pipiens in Europe. It actually pollinates the common tansy flower. Next slide. But there are other parts, especially in the United States and in Australia, where very rare orchids are pollinated by mosquitoes. So in the United States, the small bog orchid, or the Planthera obtusa, is in the northern bogs. It grows during times where mosquitoes are active, and it has been found that that plant, when it blooms, it can actually switch the type of fragrances this plant emits. I mean, imagine this. This is a plant that knows when to spray an octanol smelling type fragrance that attracts mosquitoes, and when to spray or emit a fragrance that smells more floral-like to attract either flower flies or butterflies during the day. But the mosquitoes themselves are the primary pollinators. And you can see a better picture of the pollination from that orchid in the dark there. That's an Culex communis that has a yellow wad sticking on its proboscis. And that is the pollinia, or the pollen wad, that the Platanthera obtusa actually ejects and pastes to the forehead of the mosquito. Butterflies and bees are typically too big, although they will transfer some of the nectar and a little bit of the pollen. The primary pollinator of that particular orchid is the mosquito. So when you're out and about, and now that you know so much about mosquitoes, we want you to be safe, and we want you to be able to be safe at home and out observing mosquito habitat. So when you're out, whenever possible, it is a good idea to cover up, which is easier said than done, especially if you're in the South and it's hot and miserable. But as much fabric you can put between you and the mosquito, the safer you are. And remember, it's not only mosquitoes that can transmit these pathogens. Ticks and other arthropods are out there as well. So anytime you have exposed skin, so on your neck, your ears, your hands, your arms, even your legs, if you happen to wear shorts, do wear an insect repellent. And if you're not sure as to what type to get, and you've heard all these horror stories about insect repellents that contain DEET, and you've seen the marketing in the stores that say insect repellent does not contain DEET, so therefore it's safer. Well, don't read too much into that. The EPA is helping us out and has created a tool that allows you to select an insect repellent that's appropriate for your area and for the arthropod that you're trying to avoid. And remember that anytime that you select an insect repellent, that you turn that bottle or that container over and you look for an EPA registration number, which means that that company had to do trials and prove that their insect repellent actually works, and their standard that they compare against is DEET, meaning DEET is really the golden standard. It has been developed by the Department of Defense to keep soldiers safe, and therefore the EPA uses that one to test other insect repellents against. So while I don't think I'm allowed to propose any one type, I personally use insect repellent with DEET because I know it's safe and effective. If you happen to use a permethrin insect repellent, and I've put it here in that slide, permethrin is great, but do use it only on the clothes. Don't put it on your skin, and keep it away from small children. And then while you're out and about on your own property, or when you're looking for mosquito habitat and you come across man-made containers that happen to be filled with rainwater or such, go ahead and tip and toss them out and turn them over. And then if they cannot be drained, use a larvicide of some sort to treat them. And you can also use passive autocidal overtraps to help you out further. And what those are is these right here. Next. So these passive autocidal gravitraps is a mouthful, but mainly it's a big old bucket that is usually dark, that's attractive to medically significant mosquitoes, mainly Aedes aegypti and Aedes albopictus, that can transmit dengue, yellow fever, chikungunya, and in some cases even West Nile virus. And they're attracted to these dark spaces that have water in them to lay their eggs into. And we can use that to our advantage by creating traps that mimic the same attributes. So they're dark, water filled. The mosquito enters, but it can't get out. And there are multiple products on the market, but they're in specialty stores available or through an exterminator. Into Care is one company, and also Biogens makes one as well. You set them down, they're without pesticides. The mosquitoes go in, but they can't get out. Next slide. If you come across a container that you cannot drain, be sure and treat it if you have authority to do so. Here's a couple of products that use a low-risk biolarvicide, that the active ingredient is called Bacillus thuringiensis israelensis, and it's a synthetic molecule that originated in a bacteria, and that bacteria is specific to mosquitoes and mosquito larvae and a few little tiny creatures that are closely related to mosquitoes like fungus gnats and black flies. But that is the only thing that low risk larvicide actually kills. So birds that feed on the water, that drink the water, are not harmed by it. Birds that or fish that eat the mosquito larvae that have eaten the biolarvicide are not harmed by it. So nothing else is harmed by this larvicide, only mosquitoes are. And it comes in multiple formulations. These two that I have on the screen are the retail grade. You can pick that up in any hardware store and box store. Even Walmart carries them at times. And then there's some better, stronger formulations you can get through your specialty exterminator supplier. You can treat the waters. Usually the stuff lasts about 2 weeks to 4 weeks, and you can walk away knowing that no further mosquitoes come off of that water. All right, I told you about Malaria earlier, that mosquito that's autogenous, that has a proboscis that is club shaped like a golf club. It cannot take a blood meal, and it actually doesn't want to because nature has given it really a sense of humor. So Malaria is found in the tropics and in Asia, so Borneo, Malaysia area. And it really likes to feed on sugary sap, on honeydew. But it is lazy, or it has that sense of humor, where it doesn't go and collect that honeydew itself. So what it does is it gets ants to collect the honeydew for it. The ants here, Chromatogaster, they're arboreal ants that farm aphids and scales in the tree canopies. Those aphids and scales feed on the plant sap, the leaf sap, or the leaf fluids, and those aphids produce the honeydew. The ants feed on that honeydew. And when the ants come down from the tree, the Malaya finds the downward facing ants, the downward facing Chromatogasters, and starts to harass them or tickle them or probe them with the legs and their proboscis until the Chromatogaster ants regurgitates a bit of the honeydew. So you can think of a really weird way of that Malaya feeding on ant vomit. And that is how I want to end this slide program, because I find that just absolutely bizarre, fun, and magical in a way. And I hope it also gets you to go outside, look at mosquitoes in a different way, with not just with disdain, but maybe with a sense of wonder and awe, and collect as much information as you can using the Mosquito Habitat Mapper and sending that information to NASA. So with that, I'd like to thank Dr. Scott Weaver at UTMB Galveston National Lab for sharing a couple of his biology diagrams. Cassie, our host here, that shared the video with the mosquito larvae. And I would like to thank you guys for your kind attention. If you need to get a hold of me, if you have questions, feel free to ask Cassie or get a hold of me directly at this email below. Thank you very much.
Anita, this has been so amazing, and I have enjoyed it so much. And I have to ask you a question that someone had really early on. They were asking about cryptic habitats and if there's a really weird one that you have ever found. You know, I said that when I was in Peru, a weird place that we found when I was in in the Amazon was under refrigerators. The drip pan under refrigerators was a common place to find Aedes aegypti. I was wondering if you found any weird one too.
Yes, a couple. A friend in the Houston area, so this is United States, not in a developed world, right in the middle of Houston, has a home and sent me a video of what he worried was mosquitoes, and he was right, in his dishwasher. So this was a person that didn't often use the dishwasher, and the last time they used it, didn't drain it all the way. And it was teeming with mosquito larvae. So he
Oh
was
my heavens.
freaking
Wow.
out. And so we had him drain his dishwasher. And then another odd one is in Florida, in the Florida Keys. The Florida Keys, being a chain of very small islands, early on, they didn't have plumbed water, and they used a lot of cisterns on these islands under their homes, water collecting cisterns. And those cisterns, while they're not in use anymore, because a lot of those properties now have plumbed water, the cisterns are still under the homes, and the mosquitoes breed in there. And they're actually the main problem in the Florida Keys for dengue transmission, is the mosquitoes that are under the homes that people can't get to, the mosquito control operators cannot get to.
Hmm. That's really something. I can't believe the dishwasher one. That's just
Mm-hmm.
super crazy. Another comment that I just was really interested in that William said is that in Puerto Rico, when he sees flying mosquitoes, he sprays them with just plain isopropyl alcohol. What do you think of that?
Flying mosquitoes, I don't know. I'm visualizing what that would look like spraying alcohol all over. Um, I don't know. I really don't have an answer to that. I think it probably won't do a lot of good overall unless you're walking around with a spray container with a hand sprayer, spraying every little insect that flies around you. But I don't know. That's a new one. I haven't heard of that one. So
I
I
haven't
have.
either, so I thought it'd be good to talk to you about that.
Mhm.
Okay. This has been just super fantastic. What are I have kind of a dumb question about carnivorous plants.
There's no such thing as a dumb question.
I know that. Thank you. But how is it that these mosquitoes that we find in these pitcher plants and places like that, how come they don't get digested?
Well, that's a really good question, and it's a really long answer, and
Oh,
I'm afraid we don't
okay.
have time for that. However, they have adapted over time to have extra proteins in their exoskeleton that keeps them from getting digested. So they have impermeable armor, so to speak. So they're immune to the enzymes. And the other thing on why am I a celiac
Amazing.
that I didn't even say during the program was that they can overwinter inside those purple pitcher plants
Hmm.
or inside the pitchers themselves. Those pitchers will freeze in the winter during nighttime and then thaw when the sun hits them again. And those larvae that are inside there just freeze and thaw right along the water.
Hmm.
And I see in the chat somebody mentioned that in low temperatures mosquitoes cannot survive, and I want to address that quickly. I'm in the southern United States here in the Houston, Texas area. We have mosquitoes that fly every day of the year. They're not all the same species. So different species occur at different times of the year. In the very north, there are mosquitoes that come out that emerge in February and March as soon as the ponds thaw. Mosquitoes come off the water. Those are very often Aedes. And Aedes albopictus, for instance, which is a more southern species that likes more warm temperatures, dies as an adult, but overwinters in its egg stage. So all the adults of Aedes albopictus die off in the cold temperatures. They cannot survive. So that statement is true, but the eggs survive just fine.
Yeah, and there have been some really horrific stories that we've picked up in the US where, the ambient temperature is too cold for mosquitoes to survive, but they have actually found that some mosquitoes are overwintering in parking garages where,
Yes.
where you have some buffering of the temperatures and
Mhm.
in basically all life stages, even though it's freezing outside. So they're pretty clever in finding places to overwinter.
Yes, yes. There's actually a famous population in the Northeast that is a resident population in a subway tunnel. They
Hmm.
never leave the subway. They just stay inside that subway tunnel.
Well, Anita, thank you so much. This has been absolutely wonderful. I'm really glad that we were able to run this webinar this long. And Cassie, you have just a couple announcements for us?
I do. And thank you so much, Anita. This was amazing. And I'll put the recording I'm hoping into chapters so people can jump to the different topics that you mentioned to make it easier for reviewing. So thank you for making it so easy for me to do. For those of you who joined us last month and had our conversation with Dr. Yang and now with Anita's information on mosquitoes, we're hoping that you, if you haven't already, downloaded the Globe Observer Mosquito, well, the app, and then registered and created your account, because in April, we're joining SciStarter to celebrate Citizen Science Month. And so we're going to help them as much as we can to get to their 1 million acts of or collection of science. You can connect your Globe Observer app with your SciStarter account. Both of them only need your email address to start. And if you're wondering how do you connect it, right in the app at the very bottom of the screen, there's a little gear icon. It's not showing here because I wanted to show you where you can find it. You just connect your account. Don't log out of SciStarter when you create your account. That'll automatically connect them. And then we'll be able to document all of your individual observations as a contribution to their million acts of science. So use the same email as your Globe Observer account to create your SciStarter account. We have a couple of events in April for Citizen Science Month, and one of them is how you can fight mosquito-borne disease this spring by mapping mosquito habitats. It's a SciStarter live event, and we want you to help researchers track mosquito populations and better understand the spread of mosquito-borne illnesses by joining this special webinar. It's designed for libraries and community organizers just in time for our intensive observation period, which is the month of April in Citizen Science Month. Tips and tricks and where you can go to get more information. Also in April, April 10th, GLOBE Mission Mosquito is one of 3 other apps within the GLOBE Observer app, and we have each of us will have a separate webinar in Citizen Science Month to help again with the million acts of science. April 10th at 8 p.m. is when this one will be. It's a GLOBE Observer Connect, and it is based off of a blog post that was written, and I put the link at the bottom here, observer.globe.gov. And if you just go to news and events, you'll find that mosquito habitat blog. All of our webinars are recorded, and you can find the recordings. You can find all of our webinars and recordings on globe.gov and then Mission Mosquito webinars and recordings. We have a whole host of resources that can help you if you have informal education opportunities or if you work with students as an educator. We have Beyond the Bite and Infection Detection. The Mission Mosquito Larva Hunter's Guide takes you through how to find those habitats, just as Anita pointed out. And we have a very in-depth Larva Hunters Facilitators Guide. So again, supporting you as you do some very important education work. You can earn that your students can earn some Mosquito Larva Hunter level 1 and 2 badges for completing these activities. So with that, I'm super excited to wrap this up and give you a break and to thank Anita and Rusty for facilitating this webinar for us, and thank you for attending it.
Thank you so much. It was my honor.
Thank you very much.